The activation of T cytotoxic cell is also a two-step process similar to that of activation of T helper cells, but the first signal is different as compared to the that of T helper cells. So in the first cell, the antigen presenting cell presents the antigen on the surface of MSC class 1 molecule, which is recognized by the uh, T cell receptor present on the cytotoxic T cell, and simultaneously the binding of co-receptors, in this case is CD8 cell, which binds to the alpha 3 domain of the MSC class 1 molecule. uh, Binding of both these molecules results in providing the first signal for activation of T cytotoxic cell. The second signal is results from the binding of co-stimulated molecules like CD28 to B7, that is present on the antigen presenting cell, similar to that of T helper cell signal 2. So once activated, the cytotoxic T cells then results in activation of cytotoxic T cell, which leads to apoptosis of the viral infected cells. So activation of T cytotoxic cells by binding of the MSC on molecule to the T cell receptor and the co-stimulatory molecule to the gamma 3 domain of MSC molecule on molecule results in activation of CD8 T cells, which then proliferates and differentiate into CD8 effector cells and CD8 memory T cells. CD8 effector cells then leads to production of cytotoxic T cells, and memory cells provide long-term immunity.
T cell activation is a complicated process. We will keep it simple by discussing the main steps only. We know that mature T cells are of two types, CD4 positive T cells and CD8 positive T cells. These are naive T cells since they have not encountered any antigen yet. First we will study the activation of naive CD4 positive T cells. Let's say this is an antigen-presenting cell which engulfs an extracellular antigen by the process of phagocytosis. This APC digests the engulfed antigen into peptide fragments and displays the peptide fragment on its plasma membrane. Now peptide antigen is present on the surface of antigen-presenting cell as MHC2 peptide complex. Recall that this process is known as antigen processing and presentation. These cells migrate from the site of infection to the lymph nodes, where they present antigen to the recirculating naive T cells. These cells are CD4 positive naive cells because they recognize MHC2 peptide complex displayed by the antigen presenting cell. The activation of this naive T cell requires two signals. So, T cell activation is a two-signal process. Let's see what are these signals. First signal of T cell activation. Here you can see that TCR recognize and bind the antigen and CD4 co-receptor bind the MHC2 molecule. This recognition and binding by TCR and CD4 generates the first signal of T cell activation. For full T cell activation, second signal is required. Second signal enhances the first signal and is known as co-stimulation. Without co-stimulation, the T cells which have recognized the antigen remain in a prolonged state of inactivity. This state of inactivity is known as anergy. Co-stimulators include cytokines or a pair of plasma membrane molecules. More than 20 co-stimulators are known. The most important known co-stimulator pair is B7 or CD80 and CD28. CD28 of this pair acts as receptor and is present on T cell. B7 is known as ligand and it is present on the antigen-presenting cell. These molecules also makes the two cells to adhere together for more time. Other co-stimulators include CD2, CD28, and CD45. These two signals result in the activation of T cell. As a result, the activated T cell synthesizes and secretes interleukin-2. Interleukin-2 acts as autocrine cytokines, which means it binds to its specific receptor on the same T cell. T cell undergoes proliferation and differentiation rapidly, and soon a population of effector T cells and memory T cells is generated. These CD4 positive effector T cells develop into diverse subsets of T helper cells. And CD4 positive memory T cells have ability to quickly generate more effector and memory cells if encountered by the same antigen in future. The T cell activation of CD8 positive T cells is similar with few differences, such as naive CD8 positive T cells recognize antigen presented by MHC1 molecule. The co-stimulation of these cells also occur by interleukin-2 and other cytokines produced by helper T cells. These helper cells are those which recognize and bind the same antigen. Once activated, these activated CD8 positive T cell result in the generation of effector T cells and memory T cells. Effector CD8 positive T cells are the cytotoxic T cells that attack the body cells affected by the pathogen or antigen. And memory T cells are able to quickly proliferate and differentiate into more effector cells and memory cells if the same pathogen infects in future.
So students, it's time for our second concept checkpoint. You have to make a sketch of events that takes place during T cell response to an extracellular or exogenous antigens. That is, the first step should start from capturing of that antigen, processing, then presentation, then activation of the T cells, and then differentiation and proliferation of T cells to develop its effector function. Try to find out the answer, and if there is any problem we can discuss in that in our question answer section.